Welcome back to the deep dive. If you're tracking the leading edge of advanced medical research, especially in metastatic castration-resistant prostate cancer mCRPC, keeping up feels like trying to read a textbook that's being rewritten hourly.
It really is an intensely dynamic field right now. I mean, cancer cells are just experts at developing resistance, and the scientific response is moving light years beyond standard chemo or, traditional hormone blockers.
Exactly. So our mission today is to cut through some of that complexity. We've gathered sources spanning everything from phase 3 trials to molecular strategies straight from the lab. We're not just summarizing the news, we're really trying to distill the core mechanisms.
And the overarching trend we see, the thing that unites all these different updates, is this decisive pivot. We're moving away from treating cancer based on broad classes of drugs and toward these highly targeted multi-mechanism combination therapies.
Right.
Researchers are orchestrating attacks that hit the cancer on, well, 3, sometimes 4 fronts at once: immune, radiation, metabolic, molecular, all dictated by the specific markers that tumor is showing.
Okay, let's unpack that. Let's start with the immune system, specifically a fascinating approach in immuno-oncology. Our sources highlight the ongoing phase 3 KLK2 passenger trial. It's testing a drug called Pasritamig with docetaxel chemotherapy.
And it's innovative because it addresses a major historical problem in solid tumors like prostate cancer, which is that T cells, the body's natural assassins, often can't get close enough to do their job.
And Pasritamig solves this because it's a bispecific T-cell engager?
Exactly, a bispecific.
So how does that bispecific part actually work? What is it grabbing onto?
Think of it like a microscopic handcuff. One end latches onto the tumor cell by targeting human kallikrein-2, or KLK2, a protein that's often really abundant in advanced prostate tissue.
Okay.
And the other end latches onto a T cell. So by connecting the two, the drug literally forces the T cell into intimate contact with the cancer cell, directing the immune response exactly where you need it.
And the earlier data on this, what did it suggest?
The phase 1 data was pretty remarkable, even in patients who were heavily pretreated. At the recommended dose, over 40% of participants had their PSA levels drop by at least 50%.
Wow.
And beyond that, the safety profile was favorable. Low rates of fever, no serious neurotoxicity, and most impressively, the treatment could be given without needing a hospital stay.
That's a powerful result on its own. But this phase 3 trial is combining it with docetaxel, which is classic chemo. Why bring chemotherapy into an immunotherapy trial?
Well, that's where the strategy gets really insightful. It's a strategic combination. Docetaxel doesn't just kill cancer cells. There's growing evidence that it can actually change the tumor microenvironment.
How so?
It may increase T cell activity within the tumor and release tumor antigens, which basically makes the leftover cancer cells more visible to the immune system.
So it's a classic pincer movement.
Exactly. You're attacking on the molecular and cellular levels at the same time.
I also noticed a really interesting detail about that trial, the intermittent dosing strategy. That seems like just a scheduling note, but I suspect it's more than that.
Oh, it's absolutely critical. This addresses a huge lesson we've learned over the last decade of T cell therapy. If you continuously activate T cells, they eventually just get burned out. It's called immune exhaustion.
Right.
So that intermittent dosing, spacing out the infusions, is designed to give the T cells time to rest and regenerate. The goal is to keep a healthy, functional pool of T cells that can fight the cancer long term.
Shifting gears now from the immune system to the physical precision of radiation, we've seen great success with agents like lutetium-177, but our sources are highlighting what's next. The ARTISAN, or ARPIs-001 trial, it features an alpha emitter called lead-212. This is a completely different ballgame, isn't it?
It's the move to ultra-precision. We're going from beta emitters, like lutetium, to alpha emitters. And lead-212 has 2 critical differences compared to other isotopes like actinium-225.
Okay, tell us about those features.
First is its path length. Lead-212 has an extremely short radiation path, only 40 to 80 microns. I mean, to put that in perspective, that's about the width of 1 or 2 cancer cells.
So it's hyper-localized.
And the second factor is speed. Its half-life is incredibly short, just 10.6 hours, compared to, say, 10 days for actinium-225.
Wait, 10.6 hours? That sounds like a logistical nightmare. I mean, if the drug is only active for that short a window, how do you even handle distribution?
That's a very valid challenge. You're right. It means the supply chain has to be incredibly coordinated. But that short half-life brings immense clinical benefits that, justify that complexity. The short duration means the radiation damage to healthy tissues is minimal.
So you see fewer side effects like bone marrow suppression.
Precisely. The healthy, rapidly dividing cells, like in your bone marrow, your immune system, they can recover much more quickly. This potentially reduces toxicity and gives doctors a lot more flexibility. You could give multiple doses within weeks, not months.
Which is huge for aggressive disease.
It is. And as Dr. Oliver Sartor pointed out, this rapid on and off profile makes it really well suited for new combination therapies, maybe stacking it with things like PRP inhibitors or other immunotherapies.
That's fascinating. So let's now dive deep into the molecular warfare happening inside the cell. We have some exciting preclinical news about a compound called CBPD-409, and this introduces a concept that goes way beyond just blocking something.
That's right. This is truly next-level stuff, involving a strategy called PROTAC degradation. CBPD-409 is an experimental oral molecule designed not just to inhibit a protein, but to literally remove and destroy it.
Okay, so help us define that distinction. What's the fundamental difference between inhibiting something and degrading it?
So think of a standard targeted therapy, like an RSI, as, well, it's like putting a padlock on the front door of the cancer cell's fuel source. That's inhibition. The machinery inside is still there, ready to adapt.
Right.
Degradation, using this PROTAC approach, is like getting rid of the entire machine shop. It basically harnesses the cell's own waste disposal system to tag the target protein for destruction, removing it from existence entirely.
And what specific proteins is CBPD-409 destroying?
It targets CVP and P300. And the reason is that these two act as powerful partners with the androgen receptor, the main engine of prostate cancer. They activate cancer genes through these powerful switches called enhancers.
Ah, so even when you block the receptor, these enhancers keep things running.
Exactly. By degrading CBP and P300, CBPD-409 effectively erases that enhancer signal at its source. It leads to a much more profound shutdown of the cancer's growth machinery.
And the preclinical data on combining this was promising.
Extremely. CBPD-409 combined remarkably well with enzalutamide. In models that were resistant to standard therapy, this combo led to much deeper tumor control, even regressions. It's a true 1-2 punch.
That sets the stage perfectly for our next trial, which takes molecular targeting to, well, its most personalized limit, the phase 2 POSTPONE trial, using a drug called Pacritinib.
This is the ultimate example of bespoke medicine. We've talked about general resistance, but this trial focuses on a very specific, almost paradoxical resistance loop. Standard hormonal drugs can sometimes ironically trigger the activation of something called the JAK2 STAT-T5 feed-forward loop.
Which just fuels more cancer growth.
Right. And Pacritinib is a JAK2 inhibitor designed to shut that specific loop down.
But the entry criteria sound incredibly specific.
They're non-negotiable.
Yeah.
To even get into the trial, you need a recent tumor biopsy showing detectable nuclear STAT5 in more than 5% of your cancer cells. This isn't for just any mCRPC patient. It's only for that molecular subgroup whose tumors are proven to be using this exact resistance pathway.
Hitting resistance on multiple fronts is clearly the future. Our final segment looks at an ongoing phase 1b/2 trial that's investigating a complex triple combination. It brings together metabolic disruption, targeted radiation, and hormonal suppression.
This is a fantastic example of just stacking mechanisms to overwhelm the cancer. The three parts are AMO-959 for metabolism, Pluvicto for the targeted radiation, and then standard ARPIs.
Okay, break down the role of that newest player, AMO-959.
So AMO-959 is a direct activator of AMPK, which is a fundamental metabolic regulator in our cells. The theory is that by activating it, you disrupt the cancer cell's energy engine. You basically starve it. And crucially, AMPK activation has also been shown to inhibit the androgen receptor. So AMO-959 disrupts the energy supply and potentially makes the cells much more sensitive to both the radiation from Pluvicto and the hormonal blockade from the ARPI.
So you're disrupting the engine, delivering precision radiation, and maintaining the hormonal blockade all at once. What's the current status of this?
Right now, they're in the phase 1b dose escalation stage. So the focus isn't efficacy just yet, it's safety. They have to figure out the maximum tolerated dose of AMO-959 when you stack it with the other two, before they can move on to phase 2. If we synthesize all these sources, the key takeaway is that the future of cancer treatment is entirely defined by the mechanism of resistance we're trying to fight and the precise molecular signature of the patient's tumor.
Right. It's not about broad categories anymore.
Not at all. Whether we're talking about T-cell engagement, PROTAC degradation, or shutting down a specific circuit like STAT5, the days of one-size-fits-all treatments are just. They're fading.
And understanding that unique how and why of these combination strategies is absolutely vital. We aren't just fighting the disease anymore, we're fighting the specific way that disease resists our treatments.
And that precision is perfectly embodied in a trial like POSTPONE. That requirement that patients must have detectable nuclear STAT5 in more than 5% of their cancer cells, that represents the ultimate commitment to personalized medicine. It's a promise to only deliver these powerful, complex therapies to the specific subgroup that we know can benefit.
A remarkable snapshot of science at work. Thank you for joining us on this deep dive. Stay strong and fight on.